talked a lot about different types of cell walls. Why do we care? Well, one of the biggest things that we're going to do in micro, and one of like a really powerful test is a gram stain. And so you'll guys will hear about gram positive or gram negative bacteria. So the gram stain is a differential stain, which differentiates or distinguishes cells with a gram positive cell wall from those with a gram negative cell wall. And those gram positive ones are going to be positive and retain that crystal violet and stain purple. Okay, so Prince fans, Purple Rain, it's positive, right? So gram positive is going to be your purple cells. Where gram negative, they are not going to be able to retain it, so they lose the crystal violet and they are going to stain red from the safranin counter stain. Okay, and these again are very important basics for the bacterial classification and identification, and we use it a ton in microlab. And this is a practical aid in diagnosing infection and guiding drug treatment. Okay. So there are a couple different steps to it. So the figure of a gram stain, step one is the primary dye, which is crystal violet. And this is what it's going to look like. They're both going to be purple. And then the chemical reaction occurs in the cell, and of course this is very magnified. We can't say this with our eyeballs, okay? And both cells are going to stain with the dye. But then we are going to continue the gram stain on this slide, okay? So we have the mordant or the grams iodine, okay? So this is supposed to be grams iodine, not grams iodine. So funny. Okay, and so basically our gram positive, the dye crystals are going to be trapped in the cell, where those gram negatives, it's going to wash off, and so there'll be no effect of the iodine, okay? Well then, the microscopic, then we're going to have the alcohol, which is the decolorizer, so it'll kind of fix that purple in the gram positive, where in the gram negative, it'll wash it away, okay? Because remember, it's not going to be able to stick to those. And then the chemical reaction, again, very magnified for gram positive, is going to be the blue crystals, purple crystals are going to remain in the cell, where in the gram negative, they're going to be weakened and lose that dye. So again, microscopic appearance of the cell, we're going to do the counter stain, which is the safranin, and this is the red dye counter stain. And so your gram positive kind of already filled, so the red dye is not going to have any effect, so they're going to remain purple. For the gram negative, it will be able to fill it, and the red dye is going to stain the colorless cell. So then what happens when we have non-typical cell walls? Well, some bacterial groups are going to lack typical cell wall structures like Mycobacterium and Nocardia. And so this gram-positive cell wall structure with lipid might call it cord factor. This is when pathogenicity and a high degree of resistance to certain chemicals and dyes. And this is why we came up with other staining mechanisms like the acid-fast stain. So you can use this to diagnose infections that are caused by these types of microorganisms. And some cells have no cell walls, like the mycoplasm, okay? And so the cell wall is stabilized with sterols, and it's pleomorphic, okay? So if we have a phospholipid bilayer with the embedded proteins, this is the fluid mosaic model. And this is what we talk about with the type of cell membrane that we have, okay? Ours are not rigid. So when I run into the wall, my cells don't fracture, okay? Where with a plant cell, if we bend it in half, it does snap and break, okay? And so because we have the fluid mosaic model, we have this fluid, not static cell wall, okay? And this functions to provide a site for energy reactions, nutrient processing, and synthesis, as well as the passaging from nutrients into the cell and the discharge of waste. And the cell membrane is selectively permeable. So hold on one second. Okay, so let's see if we can get this to play and record. So I'm trying to make it so that you can hear it, but I don't think that that's going to work. Okay, so look up a gram stain, okay, and we'll talk about this in lab too, but you can see how you can actually perform it. Ooh, I wonder if that works. Maybe. Okay, so look up a gram stain, okay?